Welcome, everyone. Thank you so much for joining our webinar today. My name is Susan Chung. I'm the Senior Global Product Manager for Quanta Bio. We have an exciting talk for you today on the topic of efficient and precise DNA library preparation solution for multiple sequencing platforms that are emerging in the market. A quick introduction on who we are and what we do at Quanta Bio. Quanta Bio is an innovative life sciences brand. We were founded in 2001, and we're looking to develop products for RT, qPCR, PCR, and NGS that combines application-specific enzymes with optimized reagent compositions, with the ultimate goal of combining these two aspects for superior product performance. And with that, I want to introduce to you our speaker for today. We have Dr. Subrata Panja. He is Quanta Bio's Associate Director of Field Application. He joined the Quanta team about 4 years ago, and he brings years of experience prior to contributing his knowledge to field applications. He received his PhD in biophysics. His postdoctoral research at Johns Hopkins University was to understand small RNA-based gene regulations. He also had previous work at GeneDx, where he worked on diagnostic assay development using NGS, qPCR, and dPCR.
Thank you, Susan. As Susan mentioned, today we are going to talk about rapid, efficient, and precise DNA library preparation solution for multiple sequencing platforms provided by Quanta Bio. The goal for today is to provide a brief introduction to the sequencing technology at the beginning. And for the library preparation solution, we'll mostly focus on Illumina sequencing technology and how we are providing solution there at the beginning. And then we will discuss how our solutions are compatible and you can adopt it in the new sequencing platforms like Singular Genomics, G4 platform, or Element Biosciences Avity platform. And during this presentation, I'm going to focus on few sequencing applications, including whole genome sequencing, high throughput sequencing, and some other panel-based sequencing, including whole exome or cancer panel-based sequencing. How the sequencing technology landscape evolved over time. I'm not going to go through all the details, just a global overview of the DNA sequencing technology. So around 1977, first Sanger sequencing technology emerged, and that was definitely revolutionary. Issue was it was low throughput, you can sequence only small segment of DNA. And throughout the years of development, around 1990, first capillary sequencing method came to the market, and that definitely helped generating a lot of sequencing data in relatively quick time. And that's what people normally call first generation sequencing platform. So that was good, but it was limited to the number of DNA you can sequence at a time. So throughput was not great. But 10 years later, or approximately 10 years later, the next generation sequencing technologies or second generation sequencing technologies appeared, and that revolutionized the sequencing, research, diagnostic, and everything. Two major markets that played a big role changing the sequencing landscape. Obviously, iron was one, iron torrent from Thermo, and Illumina SBS chemistries. And later, even further development happened, and starting from 2010 and later, the long read sequencing platforms came, or third generation, if you call it that way, mostly by Oxford Nanopore Technologies and Pac Biotechnologies. And this third generation sequencing technology definitely supplement or completed a lot of information missing from the second generation sequencing technologies. Now, this is a 2022 sequencing market share report. And if you look at the report, you can clearly see that Illumina is the dominating technology in the market. 80% of the market share belongs to Illumina. And then small share belongs to Iron Torrent, MGI, Oxford Nanopore, or PacBio. There are some other players too, but those numbers are really tiny. What I want to mention here is that starting from 2016, other technology companies like Singular Genomics, Ultima, or Element Bioscience, Complete Genomics powered by MGI, they are making a big impact in the market too. And the overall goal of different technologies is to make sure that the sequencing technology is available to everybody globally. Sequencing cost is lower, and you get faster, much better quality data. So, where Quanta Bio is contributing in this landscape? So, if we look at sequencing workflow, it always starts from input DNA or RNA. This talk is mostly going to focus on DNA based NGS workflows. So, again, start from input DNA and then library preparation. So, library preparation includes fragmenting the DNA, putting adapter on both sides of the fragmented DNA so that it can bind to the flow cell and you can identify the data after sequencing. And if needed, you can do high fidelity amplification to generate enough amount of DNA libraries at the end. Once libraries are prepared, sequence it using your desired sequencing platform, and then data analysis. And QuantaBio is playing a big role for that library preparation workflows because we believe if you start with good quality library, you will get good quality data. Without good quality library, it's very difficult. It does not matter what kind of platform you are using, you are not going to get good quality results. So briefly, what kind of solution QuantaBio provide? For library preparation, we have two different kits, one called Spark DNA Library Prep Kit and the other one Spark DNA Frag and Library Prep Kit, which includes enzymatic fragmentation. I'm going to elaborate all of those things in the upcoming slides. For high fidelity amplification, we have Spark Hi-Fi PCR Master Mix, which is part of both of these DNA library prep kits. And we also have Replica Hi-Fi Tough Mix recently making a big impact, and many other research groups showed that Replica Hi-Fi is probably a very diverse and universal Hi-Fi enzyme for multiple next generation sequencing applications. Spark PureMag beads for cleanup during library prep or any other kind of cleanup, size selection, and Spark Universal Library Quant Kit for qPCR based library quantification. Now, if we look at different steps of library preparation, it consists of multiple steps. So, starting from genomic DNA, then you have to fragment the DNA and repair to make sure you have perfectly double-stranded DNA. Ends are blunt-ended, and then you add a tailing step to add A at the 3' end to facilitate the ligation step, then adapter ligation, and PCR amplification. So many factors actually contribute or affect the quality of the libraries at the end. That includes obviously quality of the input DNA itself. Now, DNA fragmentation step, depending on the technology you are using, there can be bias coming from fragmentation. There can be inconsistency between different library prep, different days, and all that. And then obviously, all the enzymes involved during these steps can have impact on the quality of the libraries. And sometimes we are always concerned about the bias introduced by the PCR or high fidelity enzyme we normally use for amplification. And during these multiple steps, when you are transferring DNA from one tube to another, you can lose DNA and your yield of library can be lower. So, as I mentioned, QuantaBio has two different solutions. The first one I want to talk about is Spark DNA Library Prep Kit, which has high sensitivity and it can use a wide range of DNA input from 250 picogram to 1 microgram input DNA, and the input material is already fragmented DNA. Once the DNA is end polished, that means end repair, A-tailed, then it go through the adapter ligation step on the same tube, then you perform a bit cleanup to remove free adapters, and then if needed, you can use the hi-fi enzyme included in the kit to generate enough library for sequencing. From start to end, total time required for library preparation is 2.5 hours. Now, if you do not want to start with already fragmented DNA generated by other methods, which I'm going to elaborate later, mostly by mechanical shearing, or if you have PCR products, which is compatible with sequencing platforms, then you can use Spark DNA Library Prep Kit. But if you want to make a fully automated workflow where you want to start with genomic DNA, want to fragment the DNA with enzymatic fragmentation, then you can use Spark DNA Frag and Library Prep Kit. Against this provides streamlined workflow, wide range of input DNA, 1 nanogram to 1 microgram. As you can see, I'm emphasizing the fact that QuantaBio Spark library prep kits provide solution for wide range of input DNA. Now, in order that to happen, a critical thing is that the ligation step has to be very efficient and it should produce an equivalent amount of library irrespective of the input amount. So in this particular plot, we are showing that if you start with a certain amount of input DNA in nanogram, on the y-axis, we are plotting how much library you are getting in nanomolar determined by qPCR. And you can see that from very low quantity of input DNA up to 1 microgram, you are always getting equivalent amount of PCR-free library. That means our ligation process is efficient, it is unbiased, independent of the input DNA you are using. So bottom line is if you are starting with low quantity of DNA or high quantity of DNA, you don't need to worry about ligation efficiency of the ligase included in the kit. Now, if you don't have enough DNA, as I mentioned, then you have to amplify the library in order to generate enough quantity. And in this particular example, we used either 100 nanogram, 1 nanogram, or 250 picogram input DNA, prepared libraries, and amplified using either our Spark Hi-Fi master mix or master mix provided by competitor products, and we always get higher yield. Mentioning that when you get higher yield, that means you need to use less number of PCR cycle in order to generate a certain quantity of library. And before I mentioned, we are always worried about biases introduced during the PCR step. So what we did here, we mixed 3 bacterial genomic DNA with different GC content, which we call mixed bacterial genomic DNA. And later in the slide, I will mention it as MBGD. So 100 nanogram library input DNA was used to prepare library either with PCR-free method or with amplification. And when we looked into normalized coverage across GC content, we did not see any difference. So that clearly implies that the PCR step we included during this library prep method did not include or did not pose any bias. So which is comforting, and we can use this method to generate libraries from lower quantities of input DNA. This is another way to look at it. So we looked at normalized coverage at high AT regions and high GC regions, data generated from Quanta BioKit presented in orange, other competitor kits presented in other colors, and you can see that Spark DNA library prep kit showed minimum bias or least bias at high AT and high GC regions. And really quickly here, we are showing percentage of map reads and duplication rate, either for low input or relatively high input DNA, where libraries were prepared and amplified. You can see that map reads were high and really corroborate well with other competitor kits. And same thing when we prepared PCR-free libraries. So overall, the data you get is high quality. You get lower number of duplication reads, and map reads are very high. Not only with good quality DNA, I'm quickly sharing you some data where libraries were prepared with either cell-free DNA or FFPE DNA, and we got higher yield from those kind of difficult DNA samples when prepared with Spark DNA library prep kits. Again, saying that these kits are versatile and can be used for different inputs and different sample types. Now, I mentioned that if you want to start from genomic DNA and you want to have a streamlined workflow where fragmentation would be done during the library preparation step. So that is what we want. But let's take a step back and talk about what kind of fragmentation methods are available. So there are three major fragmentation method in the market. One is sonication or physical shearing. Second method is using transposes, commonly known as fragmentation method. And third thing is enzymatic fragmentation. Now, these three methods have its strength and weaknesses. I'm going to briefly mention it here. So sonication or physical fragmentation method is gold standard. It is reproducible, it has minimum bias, and it does not depend on the sample type. But the problem is it is expensive because you need to buy a separate instrument, separate kind of plates or device for fragmentation. High throughput, it is not high throughput, it is low throughput. And you can lose sample because you have to transfer it between different tubes or plates. But if this is your desired method and your application absolutely require using sonication or physical shearing, you can use that upstream of Spark DNA library prep kit. Now, fragmentation-based library preparation workflow is faster. It eliminates costly mechanical sharing, easy to automate. Overall, very simple. Problem is it is sensitive to input DNA amount. It introduces a lot of bias, and commonly it you lose sample complexity. Simple, expensive, and at the same time results are not that great. And finally, enzymatic fragmentations are automation friendly, high throughput, cost effective, But in many cases, that can also introduce bias, and there is a risk of being not reproducible every time. What we did within QuantaBio when we developed this Spark DNA Fragment Library Prep Kit, we kept in mind that the fragmentation technology is not introducing any bias, or we made sure it is reproducible between different days, different lots of enzymes, and so forth, so that you can have a reliable workflow. So if you look at this data, I'm going to show in this slide that the enzymatic fragmentation technology developed by QuantaBio is tunable and reproducible. So with 100 nanogram human genomic DNA, we fragmented it at different time points. And you can clearly see that as we increase the fragmentation time, you are getting shorter fragments. And that is true for different input amounts. So if you have a desired amount of insert length, you can just look at this plot and determine what is your fragment size. So you can decide the insert size you want, and you are not kind of restricted to the insert size you have to use because you are using a certain kit or fragmentation chemistry. This kind of fragmentation technology developed by QuantaBio is reproducible. Once you choose a certain fragmentation profile, you can keep that condition and use it over and over. So overall, the fragmentation profile closely resembles covariance sharing. It is tunable and reproducible, and you can have a fully automated workflow with wide range of DNA input. And I mentioned any kind of fragmentation technology may have its own bias or limitation. So we compared our fragmentation chemistry side by side with covariance sharing. So the mechanical sharing data is presented here in black, and the data we generated with our kit is presented in orange. And you can clearly see that the profile generated with our kit is very close to the data generated with covariance sharing. So we definitely provide minimum bias compared to some other kit may have higher bias, especially at the high AT and high GC regions. Now, I mentioned that our kit not only provide solution for a streamlined workflow, but it is possible to expand this technology for high throughput library preparation. So typically for a library preparation in 96 well format, which is widely available for different providers from different providers, typical reaction volumes range from 50 to 100 microliter, and maximum volume during bead cleanup or addition or after addition of beads is around 180 microliter. Now, if you want to adopt this kind of technologies into 384 well format, the main limitation is the volume capacity a 384 well plate can hold. The maximum volume a 384 well plate can hold is 50 microliter, and the recommended reaction volume is anywhere from 1 to 35 microliter. So just reducing the volume is not going to provide you a good result. So within QuantaBio, we developed a fully automated workflow that you can use for 384 well format if you need high throughput library preparation. So here is an example where the yellow one, where library was prepared from 100 nanogram input DNA in 96 well format. The DNA was fragmented for 16 minutes at 32 degree, and amplified with 5 cycles of PCR. And with 384 OL format, same amount of DNA fragmented for 20 minutes and amplified with 6 cycles of PCR, both provided or generated similar yield and similar library profile. So we have fully automated workflow. If you are interested, please reach out to us and we will be able to provide you a complete solution. Now, we always think that whether 384 OL format will give us reproducible data. So here we generated 48 libraries and we looked into the library yield. And based on our protocol, you can see the library yield was approximately 50 nanogram per microliter with tight standard deviation. At the same time, for those libraries, our desired insert size was 250 base pair, and you can see that across 48 samples, we got really tight size of the libraries or the insert size included in the libraries. So even that 384 OL format provides us very nice library yield and very nice library size profile across different samples and sample types. Now, not only for high throughput library preparation, Spark library prep kits can be used for other hybrid capture panel as well. So I'll give you just one example here. Briefly, for hybrid capture panels, we first prepared libraries with Spark DNA Frag and Library Prep Kit, and then we used a hybrid capture panel, in this particular case, IDT pan cancer panel, and then probes were bound to the libraries or desired region of the libraries, and then um, those section of the libraries were separated, and then selected gene panel was sequenced in Illumina platform. And as I said, in this particular example, XGen pan-cancer hybrid panel from IDT was used and applied towards 48 sample, and we get really nice on-target coverage and relatively lower duplication rate for across all 48 samples. And when compared with competitor kits, Spark kit provides maximum number of coverage density per library, and the data I'm not showing here. Also, the on-target coverage depth was better than the competitor kits. So overall, I just want to mention that Spark DNA Frag and Library Prep Kit and Spark DNA Library Prep Kit can provide a fully automated solution for different kind of library preparation for different sample types, wide range of DNA input, and you can use it for high throughput library preparation up to 384 workflow, and also for different kind of hybrid capture panels. Now I will move to the next section of this talk where I'm going to talk about how Spark DNA library prep workflows can be adopted in other sequencing platforms. So I'm going to only focus on Spark DNA Frag and Library Prep Kit. And briefly, this is the workflow I discussed with you so far, starting with fragmenting the DNA and end polishing, adapter ligation, amplification. First, I want to talk about our collaboration with Singular Genomics, and they developed this G4 sequencing platform. And I will talk about how our kits can be adapted for sequencing in this Singular Genomics G4 platform. So the first step is similar. You have to fragment the DNA in polishing that includes A-tailing. The adapters from singular genomics is different. You have to use singular genomics loop adapter, and as the ends are looped, then the next step will be cleaving the loop so that you have to open the stem loops, followed by using index primers, which include indexes so that you can identify the samples, and the spacer sequence S1 and S2, so that when you amplify the libraries, you have the library and at the end, which has the insert, spacer sequence, indexes, and S1, S2 sequence, which will bind to the flow cell for the G4 platform. So we used this workflow with Spark DNA Fragment Library Prep Kit. Libraries were prepared with either 500 nanogram mixed bacterial genomic DNA, 10 nanogram mixed bacterial genomic DNA, and 100 nanogram human genomic DNA. Using standard protocol, we got higher yield, good looking library. So you are happy with the libraries we generated using this method, and we sequenced it in G4 platform. So here is some data or sequencing matrices we got from G4 platform. So for both mixed bacterial genomic DNA and human genomic DNA, we got roughly 600 nanogram yield. We always target for more than 500 nanogram. Duplication rate for human genomic DNA was 6%. We always get little higher duplication rate for mixed bacterial genomic DNA irrespective of the platform. So I'll focus on human genomic DNA data. Percentage of map reads are very high, same with the percentage of reads mapped in pairs. And when we looked at the Q score, again, most of the reads were above 30%. So Q30 was very high. And we compared the data between Illumina and singular genomic DNA to look at GC content, normalized coverage across different GC content, and the data match very well with each other. Obviously, there are some difference between the technologies, but we were really happy with the data we got in singular genomics platform. Now, this is the work we did in our lab. In next two slides, I'm going to show some data published by Singular Genomics itself. So first, PCR-free whole genome sequencing on G4 platform. So for that kind of work, they used 1 microgram genomic DNA and then did coverage sharing or mechanical sharing. And then libraries were prepared using Spark DNA library prep kit, and they used full-length cleavable stem loop adapters. So these adapters have indexes and all the sequence you need. And then it was sequenced in F3 flow cells with 2 by 150 base pair format. Again, here is the mean quality score for both read 1 and read 2. And you can see that across different cycles, the quality score was above 30%. So really good quality data was generated. And when normalized coverage was detected across different GC content, starting from 10% to 70%, they observed really nice normalized coverage across or uniform normalized coverage across different GC content. You can look into this app note from Singular Genomics website. They also looked into whole genome sequencing on the G4 platform. To do that, they start with 150 nanogram human genomic DNA, and then libraries were prepared using Spark DNA Fragment Library Prep Kit and Singular Genomics Universal Adapters. So that means the indexes were added during the PCR step, And then hybrid capture was done using IDT XGen exome hybrid panel V2. And finally, sequenced it in G4 platform with 2 by 155 format. Again, when you look into the data, the mean coverage across different sample type was more than 100x. Duplication rates were less than 20%, which is good when you are doing hybrid capture. On target was close to 90%, and percentage of read aligned was above 99%. So for exome panel from IDT, they got really high quality results doing the library prep using our kit. If you are interested in this kind of technology or workflow, you can again download the app note from Singular Genomics website. So, in collaboration with Singular Genomics, we observed that our DNA library prep kits are fully compatible with their platform. And if you are thinking about acquiring one Singular Genomics platform, please try our kit in that platform. And if you need any technical help, please reach out to us. Now we want to move to the next section where we want to focus on Element Biosciences Avity platform. So on the left-hand side, this is their Avity sequencing instrument. And for sequencing, they use Avidity sequencing technology. This is really a nice and neat technology. I encourage you to go to their website and learn more about it. Now, when Element Biosciences developed their sequencing platform, they launched two different solutions for library preparation. One was Adapt workflow, where end user can use their existing Illumina libraries, DNA or RNA, and then they can use a particular kit or adapt library compatibility kit to convert the Illumina libraries compatible with the Element platform. Now that workflow is a little longer. We did not focus on that. Rather, we focused on the second workflow, which is called Elevate workflow, where you start with DNA or RNA sample. You can use our kit to generate libraries where you will include Elevate indexes and adapters. And then these linear libraries are going to be compatible with this Avity platform. Why we focused on the second platform or second workflow? Because this makes the entire workflow really shorter and easy to be adapted by the end users. So we followed two different workflows where we started with input DNA. Libraries were prepared using Spark DNA Frag and Library Prep Kit, where we used Elevate long UDI adapters. What does that mean? These are full-length UDI adapters, and the adapter sequence and indexes were included during the ligation step. And then we sequenced it in Element Avity platform with there is a type of 2 by 151 method. And we also followed another workflow where we started with input DNA. Libraries were prepared with Spark DNA Frag and Library Prep Kit. And instead of full length adapters, we used stubby adapters purchased from ITT. And you have to use UDI primers. So that means the indexes were added during the PCR step. The first workflow is compatible with PCR free library preparation. For the second one, you have to use PCR step in order to include the indexes. So here are a few examples. Either we use 500 nanogram human genomic DNA or 200 nanogram mixed bacterial genomic DNA to prepare PCR free libraries. or we use 100 nanogram mixed bacterial genomic DNA with amplification. And these libraries were prepared with Spark DNA fragment library prep kit with Elevate long UDI adapters, so full UDI adapters. And on the right hand side, 100 nanogram human genomic DNA was used to prepare libraries using stubby adapters and UDI primers from Element. For amplification, we used either Spark Hi-Fi included in the kit or Replica Hi-Fi, which I mentioned is another solution from Quanta Bio for library amplification. So I'll briefly mention you few sequencing matrices we generated or we learned from sequencing our libraries. So again, 500 nanogram human genomic DNA was prepared with PCR-free method. 200 nanogram mixed bacterial genomic DNA used for PCR-free method, and all other input DNA was used with 5 cycles of amplification. The data shaded in yellow, we used full-length UDIs, and data shaded in green color, we used stubby adapters. And this is the total number of reads we generated after sequencing. You can look at the duplication rate we got from human genomic DNA was very low, less than 5% in all cases. I mentioned the duplication rate we get from mixed bacterial genomic DNA is little higher. Again, percentage of map reads were really high, especially with human genomic DNA. It was close to 99%, and percentage of reads in pairs were also very high. So we were really happy and satisfied with the data we got from Element Avity platform. And I just want to show you two set of data which I'm particularly really excited about. If you look at the quality score or quality of reads coming from Avity platform, most of the reads had quality score more than 40%. So they are not talking about Q30 anymore. They are talking about Q40. So the confidence you get from sequencing in this platform is very, very high. And here you can see the data we generated with PCR free or amplified workflow. Both gave really high quality data. And in this particular data, I'm showing the correlation between libraries prepared with PCR or PCR free workflow give you exactly same result. So again, mentioning the fact that the amplification module included in our library prep kit do not incorporate any bias. So in last 40 plus minutes, what we learned today is no bias or minimum bias. So Spark DNA Library Prep Kit provides a streamlined solution for DNA library preparation for a wide range of DNA input, 250 picogram to 1 microgram without incorporating bias. It's tunable. If you're using Spark DNA Fragment Library Prep Kit, you get a tunable enzymatic fragmentation, but it is reproducible and it does not incorporate any bias. So the outcome is similar to covariance sharing. It can be used for high throughput library prep applications. So if you need to prepare libraries 384 at a time, we have solution for that kind of application. And also, if you need to use these libraries for downstream hybrid capture panel, this is compatible with those applications as well. And finally, our kits are compatible with multiple platforms. I showed with last 10-15 minutes that our kits are compatible with Singular Genomics G4 platform and Element Biosciences Avity platform, enabling sequencing on non-Illumina platforms. With that, I want to thank you. And if you need additional information, please reach out to me. Or you can also reach out to Susan Chung, who is our Senior Global Product Manager. So we'll be happy to answer your questions if you put your questions in the Q&A section, or you can reach out to us if you want to adapt our workflow, if you want to adapt to high throughput workflow or any other technical questions. I'll hand it over to Susan.
Thank you, Sabrata. We'll go ahead and address the questions starting with, is replica included in the library prep kits? Which one is better, Spark Hi-Fi or Replica Hi-Fi?
So I mentioned at the beginning that replica hi-fi is not included in our kit. You can get it separately. In our hand, both replica hi-fi and Spark hi-fi generates really good quality data. Even you can see from the data we recently generated in Element platform. That being said, Replica is a versatile hi-fi picture master mix. It can be used for short read platform. It can be also used for long read sequencing platforms like Oxford Nanopore or PacBio. So one answer is it is not included, but if you need a versatile solution, yes, you can use Replica Hi-Fi.
Okay, so the next question, your data suggests no or minimum PCR bias. If so, do you really need UDI in library prep?
Again, UDIs are more needed for the sequencing platform. For example, when Illumina first launched their NovaSeq sequencing platforms, then in order to minimize minimum cross contamination between different samples or adapter hopping type of problems, UDIs provide a really nice solution for those kind of issues. So it's more UDIs are more needed for the sequencing platforms rather than any issue with the library preparation, I would say.
Okay, and the next question, do you include UDI in your adapters? So I think that means is our adapters UDI, and also is UDI included in stubby adapters? So just to, on my end, UDI, our adapters are UDI, and stubby adapters can be purchased from a different provider. But Sabrata, if you have anything to add to that as well.
Yeah, so that's why I came to this slide. So we have Spark UDI adapters and Spark Pure Mag beads, which is essential for library preparation. Those are add-on items, so you can use our Spark UDI adapters and Spark Pure Mag beads. But if you need to use other adapters for some reason, like Stubby adapters, you can purchase it from other providers like ITT. It is definitely compatible with our kit.
Another question, adapters, are other adapters compatible with Spark's library prep kits?
Y- Yes, we have Spark UDI adapters, which is Y-shaped full-length adapters. You can use other Y-shaped full-length adapters with our kit. You can use stubby adapters with our kit. And if you go to our website and look into the product manual, especially for Spark DNA Fragile Library Prep Kit, we have recommendation to use different adapters as well. So short answer is yes, and please look into the product manual where we have a really nice recommendation how to use full length UDA adapters or stubby adapters.
Okay. And what is the recommended QC for PCR-free libraries?
Yeah, that's a good question. So typically, if you are using full-length Y-shaped adapters, if you run it in a tape station or bioanalyzer, you will see really weird pattern. Usually, it migrates much slower compared to fully double-stranded libraries. So Recommended QC. So for quantification, the best option is obviously use qPCR. So if you're interested, try our Spark Universal Library Quant Kit. But if you want to look into the size of the libraries, best option is to amplify the PCR free libraries between 3 to 6 cycles and then run it in a bioanalyzer or tape station to look into the size. Without this 3 to 6 cycles of PCR, if you are preparing PCR-free libraries with Y-shape adapters, you are not going to get proper size of the libraries. So for quantification, best method is qPCR, and to look into the size of the libraries, amplify the libraries and run it in a tape station or bioanalyzer or similar methods.
And what is the cost comparison of the Spark Library Prep kits with other kits? So I can take this one.
Yep.
So we do have a special pricing going on right now for the year 2024, and that could show up to about 60% savings in comparison to other library prep kits that are out in the market. And we also do have a very excellent sampling program. So if you want to test it out, and it is very cost efficient, please reach out to one of us and we can connect you to the sampling process as well as the pricing. Okay, so there is a specific question on replica. PCR with replica most often has spurious products that are large given how quickly it replicates. Has QuantaBio looked at these products and determined what sort of sequences show up in them? FYI, I have been using Replica with SQL kits replacing Kappa Hi-Fi, and I have increased yield and larger fragment sizes. It is an anonymous participant. If you want to reach out to us, I think we can talk to you one-on-one. But Subrata, if you have anything to add to that.
Yeah, so as you can see, replica is not only a hi-fi enzyme, but it is also a very fast enzyme. And I need to look into your specific application, but if you use a particular primer set and want to amplify that region, typically we recommend using 1 second per kb for less than 1 kb targets and 5 second per kb for, let's say, 1 to 10 kb targets. Now, if you are trying to amplify shorter targets and use very long extension time, replica will amplify your target. And if your primers can bind other part of the genome non-specifically, it will amplify those longer targets as well. So to get really nice band, I recommend you to stick to lower or shorter amplification time. The other thing is sometimes people get really high yield with replica, and if you somehow overload the amount of DNA in a gel, you can get a smear. So that's another possibility. But I would like to definitely learn more about the issue you are facing. So as Susan mentioned, please reach out to us and we will be happy to work with you to solve the problem you're having right now.
Okay. The last one I believe is in the library profiles. The kit produced a shoulder on the right, bigger size. How do we explain that?
I'm not 100% sure, but sometimes you see that when you have very high yield, you can see that the amount of libraries we added to the tape station was higher than what was recommended. So I'm sure if we dilute the libraries and use the recommended amount of libraries in the tape station, we may not see that. So definitely that is kind of an artifact coming from tape station results. So, but if you constant in your case, if you are seeing that kind of issues, please reach out to us and I'll be happy to discuss that.
All right. And that is it for questions that I see. There was a compliment. Thank you for nice and clear presentation. Very helpful. So thank you, Suprata. For everyone, thank you so much for attending again. And let us know if you have any questions to myself or Suprata or any of our team members. And with that, we'll end the webinar.
Thank you so much.